Waterloo Institute for Health Informatics Research, and today we're delighted to have with us Eric Jervis from the Department of Chemical Engineering. I'm not going to introduce Eric because I've got a volunteer, Jan Oshara, who's going to do that for me. Just before we get started, however, I want to bring to your attention two things. If you're interested in health informatics, we've got just a thing, maybe for some of you, it's called the Health Informatics Boot Camp. It's going to be a one-week intensive course held here at the University of Waterloo, and be sort of 8 till 5, Monday through Friday. July 18th, I guess, is the Monday to the 22nd. And you'll learn about the basics of applied health informatics. So you can take this flyer, or it's on our website, hi.uwaterloo.ca. So that's one announcement. The other announcement is our next Smarter Health seminar, which will be May 25th. We've got two great speakers coming from Smart Systems for Health Agency. They're the guys with the big bucks in Ontario to implement the infrastructure for healthcare among hospitals in Ontario. And they're going to be talking about managing risk in health information systems. So we'd be delighted if you could join us for that seminar, May 25th at 3 p.m. in DC 1302. So is there any other announcement, Dominic? Is that all right? All right. Well, without any further to do, I'll introduce my good friend, Jan Oshara.
Thank you very much. It is my pleasure to introduce my good friend and former neighbor, Eric Jervis. Eric received his degrees at the University of British Columbia. He's a die-hard Vancouverite. And he joined our university in 1998, and currently he's an associate professor in the Department of Chemical Engineering. The title of his presentation, Stem Cell Lineage and Control Analysis, and I think you will find his talk fascinating. Thank you.
We've almost got the presentation. Here it comes now. It's the last one. So as you can see, the presentation is large, and I tried to get that point across in my abstract. The help we're in trouble, we have too much data, was partly tongue-in-cheek, but part reality. My lab right now is making as much or more data than we can actually analyze properly. We are in desperate need of an informatics approach to what we're doing. We're developing it as we go. But being a chemical engineer, what we're most focused on is the growing of the cells, okay, the bioreactor in our system. But what we're really trying to do is to develop a way to make cells in culture for implantation. Okay, so when I talk about health informatics, I mean the health of a cell, okay, of a single cell. And I'm trying to understand everything I can about that individual cell so that I know that it's safe when I go to inject it into you as a pharmaceutical. Okay, now stem cells and regenerative medicine and cell therapy is going to change the world, right? That is almost a given. There are lots of examples of this happening. There are lots of examples where we're really close. Diabetes will be cured within the next 5 to 10 years. Parkinson's is already, there are some major improvements by using cells injected into a couple of regions within the brain. Heart muscle regeneration is underway. Kidney regeneration is underway. Various muscles in the body regeneration is underway. So there are many examples of where cells are being grown in a bioreactor and then injected into a patient. Okay, now there's a couple of things you got to think about, though, to put this into perspective. The first important thing is that in every cubic centimeter of my body, there are 10 to the 9 cells, about 10 to the 9 per cubic centimeter, okay? So if I want to regenerate a cubic centimeter of my tissue, okay, and make all of those cells ex vivo, in other words, in a bioreactor, I have a serious quality control problem. Because one tumor-initiating cell is going to give me cancer. Okay, so I might be able to inject I can make 10 to the 9 cells no problem in a bioreactor. Bioengineers have been doing that for 20 to 50 years, right? That's what we do. But knowing every cell and the properties of every cell in that bioreactor is something that we have never done. Okay, so in my laboratory, we call it the Laboratory for Single-Cell Bioengineering, and we mean it. We are trying to develop strategies where we know everything about as much as we can about every cell in our bioreactor from the time it goes in until the time it comes out. One of the natural things that comes out of that is a analysis or a data set that gives you the lineage, right? Because with a stem cell, we can start with one cell, goes to 2, goes to 4, goes to 8, and so on. And by monitoring and tracking every cell constantly in our bioreactor, we know the family history, which is a very big deal because now all of a sudden, all of the behaviors and phenotypes and the genotype and everything else is connected. Okay, there's a new level of connection in our data set because typically, you'd take a bioreactor, you'd make 1 million cells, you'd take a sample and you'd analyze it and you'd have some sort of distribution. But you'd have no idea how the properties you are analyzing are relative between cells. Okay? When you're trying to make the right type of cell, as I'm going to try to show, one family tree might be the right kind of cell, but if you get the wrong kind of cell early on in your bioreactor, at the end of the culture, all you've got are the wrong kind of cells. So you've got to track it in real time. You've got to be able to use that to adjust the conditions so that you make only good cells, or so that you can remove the bad ones. Okay, so that's where we're going today, okay? And I want to show you how we're doing it. I want to show you some movies of biology in action. And for some of you, you'll have never seen images like this, okay? It's really a brand new thing. So we're doing long-term live cell imaging and tracking so that we can do lineage and phenotype informatics, okay? When you look at a cell, when you go into modern biotechnology, okay, there are many things that you can measure. You can use a gene chip to do gene expression, right? And many of you will be familiar with gene chip analysis, right? Where you're actually looking at the level of messenger RNA inside the cell for every gene that could be expressed. Okay, now it's a very destructive analysis, though. You do a gene chip analysis on a cell and it's gone, it's dead. Okay, long dead, destructive. When you're looking through a microscope, okay, if you look through a microscope at an instant, the way we're normally used to looking through a microscope, you can look at shape, but you have no idea of the dynamics, okay? So lots of people are used to looking through a microscope at cells. There they are, they're just sitting there, they don't do anything, it's quite boring. Well, take a series over time and then speed it up, and all of a sudden it's quite the dance. As I said earlier, we're getting out lineage. Okay, so we're tracking individual cells. So we know every cell all the time for several days to a week or two. In other words, we take a microscope picture every 3 minutes for a week, stitch it together, and now we have the complete history of every cell in that reactor over the course of our culture. Some of you may be aware of this idea of green fluorescent protein, which has really revolutionized live cell molecular biology. Essentially, what you can do is you can take any protein of interest, fuse it to this protein called green fluorescent protein. That protein's the same reason that jellyfish glow in the ocean. And then you can put it into a cell, and you can now see where that protein is. Okay, so you can do live cell proteomic analysis. And then what's really interesting, but perhaps the most difficult to quantify, and it's way down here, and some of you might not be able to see it, is behavior. Okay? How is the cell behaving? It's a very descriptive, qualitative measure. We're working very hard to try to make that quantitative. In other words, how does the stem cell that you want to inject into the patient behave in your reactor? And how does the stem cell that you don't want to inject into a patient behave? Can you tell the difference? And if you can tell that difference, can you use it to only take the good cells? So every lab needs a hypothesis and to get funding, a rationale. So we're doing long-term live cell imaging. We're analyzing cellular behavior. We're looking at reporter gene expression during proliferation and differentiation, 2 different things. Okay, when you start with a stem cell, it can become many different types of things. So it's differentiating. Okay, and this should allow us to resolve detail and spatial and temporal dynamics of lineage commitment. In other words, take a stem cell. If I want to regenerate someone's pancreas, I want to start with a stem cell and get it to go down the differentiation pathway that would normally occur during development so that I get islet cells so that I can then transplant that into a diabetic's pancreas to cure them of diabetes. Okay, so I want to recapitulate developmental processes in my bioreactor to start with a generalized stem cell and get to a specialized cell that I can then inject into a patient and know for a fact that none of those are tumor-forming cells. Because a pancreatic tumor is a terrible thing. You tend to digest yourself from the inside out. So you don't want to do that. Okay? So this approach has the potential to answer both fundamental biological questions in differentiation, as well as important practical questions regarding cultivation and, most importantly, validation or quality control of stem cells that we want to implant into people at a single cell level. So what do? Here's a microscope image that you might see looking through a differential interference contrast microscope. Okay, so a high-grade, research-grade microscope. Digital camera. Now, if you do this in time, you get a sequence. Okay? And then you hire some outstandingly talented and dedicated co-op students, and they can turn this into this. Okay? So in other words, they're watching the movie and tracking manually every frame over the course of a week or so. Dedicated students, their names go on papers as they get published. This is not fun. However, our visual cortex is extremely good at this, and computers are terrible at it. Okay? Once we get all this information, the other thing that the students are doing is they're extracting phenotype and behaviors and putting them into data tables. Here's the informatics part. Okay? So we take an image sequence, track it, assign phenotypes and behaviors, enter it into a database structure so that we can now query this database. In other words, show me all the cells in my culture that behave like this. And it would light up as a red dot on this image sequence. And now we could look at those cells that were fitting our criteria when we queried this database. Okay? Real simple. So here's an example of the sort of data we get. So this is a neural stem cell cluster. It shows really good here. This is why I'm buying my own computer. My guess is this is the bandwidth from here to there is not good enough. I'm about to. Okay. You know, this is insane. What are we looking at here? Let's you guys over there. You know, this is difficult. So this is a large movie. One down. Yeah, let's get friendly here. There's nothing we can do about this. It's important that you see the sort of resolution so you know what I'm talking about when I say behaviors. Okay? Yeah, I think you've seen this one, but we'll just sort of get in here. And I apologize for this. I'm going to have my own computer within a week. This is the second time this has happened to me in a very short period of time. Okay? So what you're watching here, okay, is this movie of individual cells behaving. And as I say, I apologize for the lack of computing power here. I mean, this is Waterloo, right? So there'll be some other ones, and ideally one or two of them might come up onto this big screen. Let's see how that goes. So, yeah, and then as soon as the movie starts to go, it's gone. Your computer system bites. Okay, so what are you watching now? So we track all of the cells over time, okay? And when this goes gray, what that is the final DIC time course image, okay? And when this goes to color, what we're doing is we're using antibodies and immunofluorescence to allow us to identify the types of cells we have. So we started with neural stem cells. We put them in our culture chamber. We tracked them for this culture is about 4 or 5 days. Then we fixed and stained it and imaged it on a fluorescence microscope so that we know who's who. And in this particular case, the red ones are neurons, the green ones are astrocytes, and we believe that the darker ones in here are oligodendrocytes, although that staining is inconclusive. They are not neurons or astrocytes. In other words, we know the eventual cell type now from our lineage. So what do we have now? We have a family tree, and we know what they became. Okay, now a year and a half ago, 2 years ago, 3 years ago, I got up at an NCE meeting, a National Centers of Excellence on Stem Cells, and I said I could do this. I'm going to make a movie. I'm going to start with one cell. I'm going to make a movie. I'm going to figure out who's who. Now I'm going to run the movie backwards, okay, so that I can identify the potentiality of every node in my family tree. Okay? So if the daughters below a particular mother can become a neuron and an astrocyte, that was a bipotential node. If the children of that mother could become a neuron, an oligodendrocyte, and an astrocyte, that was a tripotential node. Okay, so now we could identify the potentiality of every cell by running it backwards, and then what we do is we run the movie forward again, so now we know what we're watching. Okay, in other words, when we're watching the cell, we know that's a bipotential cell because eventually it will give rise to two different types of children. Except now we can feature extract to see what a bipotential cell behaves like. Okay, and the Stem Cell Network NCE gave me a lot of money to do that, or some money. No one gets a lot of money. And some of the major players in Canadian stem cell research joined the project because they've never been able to do that because it's technologically challenging in many ways, both at an informatics level, a chemical engineering level. There's a lot of software development that went into this, okay, and at a biological level. What does it mean? And there's not a lot of labs, certainly in Canada, that can bring together all the technology and all of the cell biology to try to make this function. So once we do that, and there'll be other movies here, and as soon as I get the movies working on a real computer, I invite you all to my office and you can come and see it. I'll hand out my card later on, because this stuff is worth seeing. But what does this mean now? Okay, so remember, at the end of the movie, okay, we fixed and stained them with color. So we have yellow, which is our oligodendrocyte or our unstained, red, which is a neuron, and blue, which is astrocyte. Okay? So up at the top there is a cell that's got three colors on it. That's a tripotential cell. Okay? And then that tripotential cell had two tripotential daughters. That was a symmetric division. That's a big deal to stem cell people because what just happened? We now have an extra stem cell, right? One stem cell went to two tripotential stem cells. We've expanded the stem cell number. That's a big deal. This stem cell went through an asymmetric division to give a tripotential and a monopotential. Okay, in other words, all of the children from this cell only became blue, or were only blue all the way through. Here's a tri going to a tri and a bi. Okay? So that's the way we work this. And so when we're watching the movie later, this is good. This is bad. So it can't show my movies at all.
Eric, why don't you invite people up after?
Yeah, we'll do that for those. I mean, this one it won't even show. So this shows on a real computer. Yeah, I'm at Waterloo here. Okay, so in this movie that you can't see are a bunch of cells dancing around with pie charts on them. So we know what they can be. Okay, so at the start of this is two cells with two tricolor pie charts on them walking around. So the person watching that movie knows that they're watching two tripotential cells. And eventually you watch them divide, and now you can watch, well, what exactly happens when you have an asymmetric division where a tripotential goes to a tri and a mono? Was there anything characteristic of that division in terms of the way it looked that we can make use of? Yeah. Okay. So it likes the static pictures just fine. So I can use this to sort of walk you through one of the other experiments that we did. So here's another cluster. Okay, now this is actually kind of an interesting idea, and this is where we're really trying to go here. So in this particular neural stem cell cluster, we could routinely observe cells with this phenotype. Okay, now we called it the free phenotype. In other words, this cell is free from the main disk. Okay, it always sort of walks around the edge, so it breaks the boundary. And so there's some other ones down here that basically have this kind of phenotype as well. And we could reproducibly recognize that with our eyes. Okay, so let's do a lineage tracking. So here's a lineage tree. Okay, as we're doing the tracking, we're filling in those data tables. So we have all these properties. Okay, so if we zoom in on part of the lineage tree, here's one branch in the lineage tree, and we're displaying numerous properties on here. So there's an informatics thing going on here. In other words, we're trying to visualize behavior, okay, on top of a lineage tree. Okay, so this is like a multidimensional visualization of a lot of information, okay, because in this direction is time, okay, but we have all the connectivity because of the lineage tree, so we know how the mother is related to the properties of the daughter, and so on, okay, all the way down. So we could do things like light up all the cells in our lineage tree that have this behavior. And then that can be output into a lineage tree representation, and we can see, here they are, here they are, and here they are, and so on, okay? So now we can go back and forth between the observation and the lineage tree and look at what's going on. We can also make other queries. For example, show me all those cells who spent a certain amount of time near one of these cells. In other words, do we have local microenvironmental imprinting? Does a cell that tends to spend a lot of time near a free cell turn into a free cell? Okay, so it's a good question, because now we're getting at local microenvironment. And cells make decisions based on the local chemical environment. Okay, so the hypothesis that we were testing here is, does the time spent near a free cell determine whether or not a cell will become a free cell? In this case, no, it didn't correlate. But in other examples, that would correlate, because these cells would be making specific factors that would be signaling to their neighbors. So we would expect other types of effects to occur. Okay, but that's just one example of the way that this technology can be used to look at the relationships in the lineage. We can do all sorts of tracking. We can build up large amounts of data, and now we can start doing cross-plots and all sorts of analysis. For example, here's a cell cycle time histogram. So this histogram is created by looking at how long it takes every cell to go from when its mum divides until it divides. Okay, but because we're looking at the cells every 3 minutes, we really have an accurate measure of the cell cycle time. Right? This is cell cycle time plus or minus 3 minutes. And then we can make histograms. Well, what's the first thing you notice? We've got two populations. Right? So even though we started clonally, okay, we started with only, in this case, no, this case started out with more than one cell. But we can recognize two distinct populations here in terms of cell cycle time. So if eventually we were able to correlate implantability or long-term regenerative potential with cell cycle time, we would know we want these guys, we don't want these guys. Okay, so there's a feature we can pick out. We can do plots of cell cycle time versus when they divided versus how fast they go. Okay, the size of the dots here are the speed, and you can just visually see that they separate out into clusters. And this is no principal component analysis, nothing like that. All we're doing is just raw data plots to see whether or not things separate out so that we can start to get them into clusters and classifications. Okay, so that's sort of an informatics picture. But I wanted to give some more examples of the kinds of data, the kinds of things we're doing. Okay, so the last set was all neural stem cell. Okay, so these are neurosphere-derived cells. They're primary cells derived from a mouse. We know they're stem cells because they're derived from a neurosphere, and essentially the non-stem cells die off when they go into culture, and then we watch them do their thing. Well, you can do the same thing with hematopoietic stem cells, but things get way more interesting because they're easier to work with, and people have been doing blood stem cells for a long time, right? Bone marrow transplants, that's all that kind of stuff. So this is a really, I like this experiment. You'll like this one. So, well, anyway. So in Vancouver, someone will take 20 mice, okay, and they will be sacrificed, and the bone marrow taken from those mice run through a big separation process, and we're going to get a few hundred cells, okay? But those will be 40% hematopoietic stem cells. Okay? So in Vancouver, they put them into a little tube. So we've got some 20-micron spheres. We've got about 100 of them in a tube in Vancouver, FedEx overnight to Waterloo. Okay? We then find all of those cells, and that's a trick in itself. Think about it. We've got 120-micron spheres, and they're very expensive, so we want to find as many as we can. We then lineage track them, collect up all the cells at the end of each lineage, put them into tubes. So one tube comes out, and we ship back 60 or 70 tubes. Okay? Each one of those tubes then gets injected into an irradiated mouse. Why? Well, with the irradiation, we just wiped out the mouse's complete hematopoietic system. In other words, it's got no blood system anymore. So if there's a stem cell in the cells that we inject into the mouse, it lives. In other words, it's legs down. If there's no stem cells in the sample that we inject into the mouse, it passes away after 20 weeks. So we have a definitive readout at the end of our lineage, okay, whether or not we had a real stem cell there. Okay? So we do our lineage, we start with one cell, we make our lineage, we take that, pool that from one lineage back to Vancouver, they inject them into mice, and we can see whether or not the mouse lives. So the hematopoietic system has a definitive readout, the competitive repopulating unit. Okay? In other words, if this cell will repopulate that hematopoietic system of the mouse, it was a stem cell. Okay? So all cells resulting from one initial starting cell are injected into a mouse. We do the assay, and we're looking to see if the mouse repopulates over the long term. Okay? Then the question is, can we predict competitive repopulation activity based on lineage and tracking analysis? Okay? Will this allow us to identify true hematopoietic stem cells and their progenitors within lineages? Okay? And again, remember, the data analysis retains the hereditary information, so we can do mother, daughter, sister, sister, cousin, cousin, and so on comparisons. Now, why do we want to do this? Here's the deal. People have been working in mice forever. Okay? We know the molecular markers that define a hematopoietic stem cell in a mouse. Pretty close. We can do this big sort on the mouse bone marrow, and we can get out, so we've got a pretty pure population. We don't have those markers with human. Okay? We do not have a well-defined set of molecular markers that allow us to pull out a hematopoietic stem cell from a human sample. We can get it, the best we can do is 1 in 500. So, can we take the information we get by watching the cells and classifying and do cluster analysis, and then take that information and apply it to a human system? So now when we're watching, we can say, aha, that one, and that one are your true stem cells. The other cells are not the ones you're interested in. So rather than molecular markers, we're looking for phenotype markers, because the molecular markers don't exist. Okay? All right. Okay, so here we're scaling up our lineage tracking. Okay? So we've gone to lower magnification, but now we have an array of wells where we're starting with one, so this one's already divided. So we're starting with one cell, and it's skipping frames because the video card bites. Essentially what we're doing is we're doing a whole bunch of lineage trees at the same time. Okay? We're in the process of trying to scale this up in a big way because, of course, to apply this to a human system, we probably have to do several thousand lineage trees. But it's all software once you got it worked out. So this, it runs well as it's loading into RAM, and then it runs out of RAM. So this is massively sped up. You're looking at a movie that took 4 days to make. But you can see we're doing lots of lineage tracking. Okay, there's lots of lineages here. So we can take that information and, okay. So we make lineage trees. And because we're in sort of a formative time right now, what you're looking at is actually a printout of all the lineage trees. And then we went through and hand with a highlighter color-coded our lineage trees. And then you stand back and you go, what do you see? Okay? Because the best pattern recognition engine on the planet right now is right here. And we're trying to make use of it if we can. So here's one particular run. Okay? What are you looking at? Well, we have about 65 lineage trees in this top panel. Okay, so this is sort of a low magnification view. And if we zoom into this region up there in the corner there, that's here. So what do we have? We have a family tree here. And what we've done is we've color-coded, okay, the cell cycle times depending upon how that cell cycle time relates to the average cell cycle time at any particular level in the lineages. Okay? So if it's 1 standard deviation, then longer than the average cell cycle time at any particular level, we color it green. If it's 2 standard deviations longer than the average cell cycle time at any particular, at that level, we color it blue. Okay? If it's 1 standard deviation faster, we color it pink. Okay? The lineages that gave rise to mice that lived, okay, are these ones with the square boxes over the top, essentially. Okay? And so what we're looking at now, and we're trying to get more and more data to make this more concrete, in other words, we're trying to do more lineages and pick out more phenotypes, is looking at, can we, by this pattern of long cell cycle times, recognize repopulators? At a first cut, absolutely, in the sense that anything with pink in it will not repopulate right away. Interesting thing about that is most of the cells at the end of this culture, okay, will arise from lineages that have the pink because they're the fastest growing cells and they're growing exponentially. So I was speaking with a colleague in Toronto the other day, and he's used to doing 1-week cultures. So he puts one cell into the well, and then he comes back in a week and he says, yeah, they all have about, a few thousand cells. But he's not looking at every division. Because what's happening in his system is he's got these slow dividers, but as soon as one of these pink guys emerges, very quickly overwhelms all the other cells because he's growing faster. So if you're trying to just make stem cells, as soon as you see one of these, you want to remove it from your population because it will very quickly overgrow. In other words, almost all the cells, by the time you're done, will be fast-dividing cells which do not repopulate. Okay? Other things that we're starting to see, and again, this is preliminary results, is that if you maintain longer cell cycle times across, horizontally in a lineage, you have a higher probability of that being a repopulator. Okay? So it's not enough to maintain one long side and everything else accelerate, but you need to have some on both sides. So far, we need more data, okay? We're doing cluster analysis, pattern recognition, but that would be an extremely useful pattern because then we could take human umbilical cord stem cells. We got tons of it, right? Every child that's born has an umbilical cord. That umbilical cord is full of stem cells. But we can't pick out the stem cells because we don't have the markers. But if the human umbilical cord stem cells behave the same way as the mice do in culture, we could very quickly get rid of most of the cells and purify our population. Because every cell you inject into a patient has a probability of giving rise to a tumor. Okay? One of the things that you may or may not realize is that a human embryonic stem cell, if I inject it into you, you're getting a tumor. Right? You cannot inject human embryonic stem cells into someone because they're tumorigenic. Okay? They'll give rise to tumors everywhere in your body, essentially. You've got to inject progenitors. So you've got to be very careful of the level of differentiation that you're injecting into the cells, or that you're injecting into the patient, rather. Okay, so this is a hematopoietic system. You can see what we're trying to do. We're trying to recognize features in the lineage, and really, unless you're tracking every cell through every division, there's no way else to do this, right? You've got to watch it. Because if you just come back and look at it every so often, you have no idea who's who. Okay, another example. And so now we're starting to get into GFP markers. This example is highly relevant to gene therapy. So far, gene therapy has been very good at killing people. Okay, so there's a serious problem where the gene gets integrated into the genome of the recipient cell. Okay? For example, over in France, they found out there were chromosome translocations. All those kids that died of leukemia, the 3 or 4 boys that died of leukemia from a gene therapy trial, essentially the retrovirus went into the genome. There were some rearrangements, gave them leukemia. Not a good thing. Okay, so we really need to understand what the gene is doing in the host genome, how stable it is, where is it, what's it doing to the behavior of the cell, are we perturbing the behavior of the cell? So in other words, you want to put the gene into the cell in ex vivo, characterize it and quantify it and know what it is, and then you inject it into the patient. Okay, so that's what we're looking at a model for that here. So transgenes are often subject to partial gene silencing. In other words, you take an embryonic stem cell, you put in the gene, it's expressing it very nicely, you hit the stem cell to differentiate, the gene turns off. Because the chromosome, okay, the chromatin goes from active to inactive chromatin. Okay? In other words, it switches it off. Okay? And it can switch between the silent and expressing states as well. In other words, it's an unstable expression pattern. Okay? And retroviral vectors, which are a good way to get DNA into a cell for gene therapy, are subject to variegation in ESCs, embryonic stem cells, and this may also be true in adult stem cells. Okay? So there's a need to understand the stability of expression in these transduced vectors. Okay, because variegation will have important implications for things like gene therapy, for things like using genes for studying promoter regulation, and looking at various aspects of stem cell biology. Okay, variegation, okay, this switching from an active to a silent state is an epigenetic effect. In other words, it's happening with a modification of the genome, and it's inherited by its kids. Okay, so if my mom silences, I'm probably going to be silenced too. So there's a hereditary aspect to gene silencing. In other words, it's a perfect application of lineage tracking because we know that there is an inherited effect. So we should be able to pick that up in our lineage trees. Okay? And really, there's no other way to do this because unless you know the connectivity of all of the individuals in your population, in other words, the lineage tree, you have no idea how the silent state actually starts to dominate your population. Okay? Standard analysis or FACS analysis just gives you a population readout, and you have no idea of the relationship between cells in your system. This one, surprisingly enough, plays, and it's one of the largest movies. I don't know. What are we looking at? Embryonic stem cells, 2 colonies starting up in our field of view. Every time it goes black, what we're looking at is the expression level of our transgene. Okay, so it's a GFP transgene here. And so we can go through, and what are we doing? We're tracking all the cells. Okay, so we know where everybody is. And then every time we do a fluorescence measure, we can quantify how bright it is. And so now we know how active our transgene is. Okay? So this is an embryonic stem cell colony. Still growing. You can see that not all the cells are glowing. Okay? So some of the cells have silenced our transgene already. Those would not be good cells to inject into a patient. This is essentially the kind of detail that we can see. And on a smaller screen, you would see it very, like on a good monitor, you can really pick up the detail. So this culture lasted about 4 or 5 days you're watching here over time. So there's maybe 100 or so cells there. Just so you can get a frame of reference here, a cell is about that size. The 2 little blobs there, those are the nucleoli. Okay, so we got that one. This is, I have no idea why I played that one. That movie's huge. Okay. So, what did we do? We had DIC images, which we can track. We had fluorescence intensity. Okay. Here's our final population, and they're color-coded based on who the founding parent was. So we had essentially 6 major lineages here. Okay, so each color, okay, represents the children of one particular founder cell. So we have the family tree. Okay, so we made a bunch of lineages. Here's our lineage trees that we made from that movie. Okay, so we've got our family trees. And the dots here, the size of these purple dots, is proportional to how bright the cells were. So it allows us to visualize the variation in our transgene expression across our lineage. Okay? And we can do things like, how does the heterogeneity develop in time? So here's an interesting plot that allows us to get at part of that question. What are we doing? Well, here's the cell fluorescence versus their sister. Okay, so my sister's brightness versus my brightness during my lifetime. So you can see that they lie on a 45-degree line. So in other words, most of the divisions are symmetric with respect to transgene expression level. Okay, so I'm probably going to express about the same as my sister. Okay, here's the self versus the mother. So you can see that deviation is starting to arise. In other words, cells are starting to get further and further from the 45-degree line. So there's divergence in the expression level as you go vertical in the lineage tree. In other words, I'm more similar to my sister than I am to my mother. Okay? Here's the grandmother plot. So my grandmother and the self. And again, it's even further off. Okay? So although the divisions are symmetric, deviations accumulate as you go through time. And then this is just a random plot where it's one random cell plotted versus another random cell to show that it's nowhere near the 45 degree. Okay, so there's a lot of variation in our population. In other words, that's what that's saying. Okay, so why would this be of use? Well, some very interesting observations came out of this. Here's the first one I think is kind of the most interesting observation here. So we started with one cell right here that divided into two. So what you're looking at is time, intensity, and each line, each individual line is a cell. Okay? So at this point here, this cell divided into two, right at that point there. Okay? And then it continues along and, looking at the various colors, you can see that there was a big division set here. There's divisions up there. So every time a line bifurcates and goes into two, there was a division. Now what's interesting here? Well, there are 4 or 5 cell cycles across here, okay? But right around here, at 70 hours, everyone's silenced simultaneously. What does that mean? Well, it means that the signal to shut down must have been set up at least this far back. Right? Because everyone maintained that signal during the next, so there's a division, and then somewhere in here, for some reason, everybody says shut down. We've gone through enough divisions now, shut off. These would be terrible cells to inject for gene therapy because they wouldn't have any effect. So that was a very bad clone, okay, in the sense that for a gene therapy application. But this is the problem with variegation. So there was a signal that was initiated at least this far back, and it could be further back, that there will be silencing coming up, and essentially all the children silenced simultaneously at around 70 hours. So there's an epigenetic trajectory, okay, an expression trajectory that's maintained over multiple cell cycles, which is consistent with biology because differentiation and these sorts of things can happen over multiple cell cycles as well. But here's a quantitative measure of how robust that program is. Here's another interesting one. There was a bifurcation event that really happened here. So all of the children from this guy are dotted, and the children from the other one are solid. And you can see that they really sort of fall into 2 basic classes. Okay? So again, this was a asymmetric division. In other words, there was a bifurcation there in which the 2 sisters had very different outcomes, okay? But then after that, everything stays fairly consistent, except for guys that really fall off the program, and they actually die. Anything that terminates before it gets to the end is a cell that died. So this cell that was not in the family program is on its way to death anyway. So it's initiated a whole other genetic cascade that had nothing. So this particular expression level was probably just being swept along with the cell death program that's been initiated. Okay, but again, we can see that there's an instability here, and that even though we start at an expression level here, when we're done, we have expression levels all over the place. So if the amount of gene expression was important for this particular gene therapy, this would be a real problem. Okay, and you'll notice that there are no actual successful gene therapy trials continuing on right now. There's a bit of a moratorium on them because putting things into the genome is a lot tougher than it looks. And there's a very interesting, there were a lot of cowboys, and a lot of my colleagues at the hospitals call them cowboys, that were trying gene therapy way back. Right? Let's get the genes in there, let's see what happens. And they ended up killing people because we really didn't understand the process. Now we're going through the same kind of thing with stem cells. Okay, people are very interested in getting those stem cells into the patient. Okay, because there's commercial issues, there's saving the world issues, everyone has different sort of motivations. But we better be sure we're not getting ahead of ourselves, because quality control is a serious problem. 10 to the 9 per cubic centimeter. So if you want to regenerate a serious chunk of muscle, you're putting in a lot of cells. So you have a reasonable probability that you're going to have some problems. Okay, so we come back to this. We can do a lot of stuff. So in time, so right now, we're already doing this, and had you been able to see some of the more high-resolution movies, you would see that we can really see a lot of detail. I had a student working for me, a co-op student who was extremely creative 2 years ago, And she likened the behavior of the cells to a cocktail party. So she had minglers and wallflowers. So she anthropomorphized cells, but that's how she was able to keep track in her head and input it into the database. So we could say, okay, show us all the wallflowers in our population. So this was just her way of trying to put down in some way she can write down these observations, okay? And this is where we're at right now. We're trying to recognize behaviors, okay? It is very difficult because this is something we do sort of all the time, but a computer is terrible at. So we have a serious information problem. The lineage comes by doing a frame every 3 minutes over several days. That is technically difficult. Okay, so being able to stay in focus and healthy cells, right? One of the things that engineers often get trapped into is doing a lot of analysis on cells that are sick or dead. The cells have to be good first, and then you do some stuff. In other words, the biology has to be perfect before you can do any engineering, or you're not engineering anything. So we take special care to make sure that our cells are happy. We're working in proteome in a variety of ways. I've got people now expressing different proteins so that we can do these juxtaposition analysis. Because we can do the lineage tracking, if this cell is making something special and we put a cell beside it, can we pick up that interaction? In other words, can we use cells to engineer the microenvironment of our target cells? Okay, so we can do that now. It's just genetic engineering. Behavior, as I said, is something that's really, really tricky. And in a new program that's coming up in the NCE, we're going to be, one of the readouts, okay, at the end of our lineage will be gene chip expression. So we'll be essentially doing single or 2 or 3 cell gene chip experiments where essentially these guys from this lineage would be compared with these guys in this lineage at a gene chip level to see if we can pick up this variation. Okay, so we're starting to go more molecular as well. Now, I like to show this picture, and I'm glad it shows, this is one cell. So anyone who does biology says, okay, yeah, there's one cell. Now watch the chromosomes. Again, this is one cell. There's the chromosomes. They're trying to line up. It wants to divide. That didn't work. Let's try this. Those are chromosomes. So that one cell just quadruplicated. Now find that in a biology textbook. Now we've seen that in all the different types of primary cells that we toss into our system. In other words, if you look at enough cells, you're going to see some really weird stuff. And I like this movie because watch if you watch the chromosomes, they go into the Mercedes-Benz sign. So somehow the cell has a mechanism for recovering from this particular state, which is, for me, that's pretty cool, right? How often does this happen? Think about the way the DNA is partitioned when you have a triplication or a quadruplication, right? It's not each sister getting an equal pair. Each one is sharing half of its DNA with one of its sisters. So you have a really bizarre way of sharing genetic information here. Okay, so these cells are messed. But that's a primary neural stem cell. Or at least it was derived from, it's a primary cell that's been in culture for less than 5 or 6 passages. Okay, so it's a primary cell. But it just quadruplicated. I wouldn't want to put that into a patient. Okay? So let me put this into perspective. We want to make stem cells so we can do regenerative medicine. Okay? Bioengineers can grow cells in a bioreactor. We can make tons of cells. I have colleagues making vats of stem cells. But in that vat of stem cells, if you've got 10 to the 6 cells per milliliter, and you've got, say it's a liter, you've really got 10 to the 9 individuals. You have no idea which cells are good and which cells are bad because you don't know the complete family history. You have a limited number of molecular markers that you can use to analyze your population. Okay? Here's what we're trying to do. State of the art is using antibodies to look at cell surface markers to tell what the cell is. Even with a really good flow cytometer, okay, and lasers, you can maybe do 5 or 6 colors. Okay? So that means that your property vector has a dimension 5. If you do high-resolution DIC images and you get really creative people to look at those pictures and you get really creative image analysis people to try to feature extract, the size of the property vector can be very large. 50 or 60 features could be extracted for each cell from those images if you went in there and really extracted the features. Now when you do pattern recognition and cluster analysis, you've got a serious amount of data, and you can get these into small enough clusters. So you can start to say, this is a specific set of those 50 features that we're looking for when we want to inject a cell into people. Okay? So we're trying to go sort of back in time, and we're being very descriptive. We're sort of undoing all the molecular biology, and we're saying, get a microscope and look at your cells, characterize them that way, so that you have an individual single-cell level of quality control and quality assurance. So that when you put it into your patient, you know what you got. This work is done with a lot of people, and this is a partial list. Cindy Mooreshead has been supplying the neural stem cells and working with us on this project from the start. James Ellis supplies the embryonic stem cells. Connie Eves in Vancouver supplies the hematopoietic stem cells. We have a collaborator in Montreal who supplies islets, and so on. So I'm really a core imaging facility in Canada, and all of the biologists are sending us cells. A large number of students have been through. Some Heathers are in the lab right now. Dwayne has just joined the lab. Kristen, Liam, Meredith, Aaron, and a few others have done a ton of tracking. I'd like to acknowledge their contribution to all of this work. Okay. April Blaylock is a Mech-Eng student. Okay, so I've got a mechatronics student who's now going to start looking at how can we put this cell beside this cell. It's really a robotic problem, especially if you want to do it 10,000 times. And, of course, John Ramunis has been involved in all of this, software, hardware. Give him an impossible problem, and next week it'll be done. And funding, we have funding from NSERC in the Discovery Grants, the Strategic Grant Program, NSERC Cell Network, Photonics Research Ontario, and this is the big one. National Centers of Excellence on Stem Cell Network have supplied most of the cash for what you've seen today. So at this point, I'd like to stop talking and hopefully entertain some questions. Anyone?
Questions or comments?
Okay, I got one. Come on, you guys. Is this real? Are you really trying to do this? This is small-scale stuff. We're trying to lay the groundwork. A question.
Don't you worry about the subjectivity between people who are observing the cells and other behaviors that you're observing and how consistent are they between them?
Yeah, so the question is, how subjective is our analysis? With people, it's very subjective. But what we're trying to use the people for is to train the computer to classify. If a person can't recognize it, a computer is going to have a heck of a time. Okay? The visual cortex of the humans is extremely good at recognizing patterns. Computers are crappy. Okay? So what I'm trying to do is develop a whole bunch of subjective characteristics. We'll see what hangs, and then we'll try to train a computer to see the same thing. And then it becomes more objective. Right? But we really don't know what we're looking for. No one's done this before. I mean, this is really brand new stuff, right? I mean, no one has done long-term lineage tracking in a bioreactor context for quality control, quality assurance with stem cells. How can I put this? With all due humbleness, the Toronto Star had a special on how Toronto is the most amazing place to do research in the world 2 weeks ago. Front page story. Toronto Star, front page. On the front page, they listed 4 of the superstars down in Toronto that are just leading the way. 3 of those people are on this project, okay, that I'm PIing, because they recognize what we're doing as being fundamentally new and gives them a whole new direction for collecting data. Okay, so it's new.
A question about some of the data that you're
Sure.
showing with the cell cycle times, and you have
Right.
the pink lines and the yellow lines,
Right.
and the repopulation assay. Now, the pink ones that were dividing very rapidly failed in that assay.
Yes.
Were they still stem cells, or something
No.
you had mentioned that if you put?
They had differentiated.
Okay, and yet they were still dividing rapidly.
Our blood system repopulates every day. So the blood system is, in fact, highly proliferative. Right? We're redoing, I'm not sure, I think it's like 20% of our blood system every day. So there's a lot of turnover. So as the hematopoietic system cells differentiate, they start speeding up. Okay, so they overgrow very quickly.
And just a more general question to reveal my ignorance. How many different types of stem cells do we have? And how many can you recognize?
No, I mean, that's a deep question. That's a very non-trivial question, because stem cell, the classic definition of a stem cell is a cell that has multipotentiality and can self-renew. That's a stem cell. So, but then you have to think about it. Multipotentiality, how subtle of those differences will still mean a stem cell? You know, are we talking about two different types of neurons? You know, that could be a neural stem cell because it can become two different types of things, and maybe it can self-renew at that spot. So the number of stem cells, different types of stem cells, tons. Now there's one embryonic stem cell. Okay, so that special embryonic stem cell, if you implant it into a developing embryo, will give rise to all the different tissues in a body. Okay, now it's going to give tumors everywhere too, but it will be absolute pluripotent. Okay, so here's the thing. A fertilized egg is totipotent. It can give rise to everything, okay, including the placenta, the extra embryonic tissue. Not all of the placenta, but the part of it. Then you've got totipotent that can give rise to, totipotent, pluripotent that can give rise to all of the cells actually in the organism. And then you've got all the tissue-specific stem cells that can give rise to a specific tissue. And then you've got other stem cells that can give rise to different types of cells within the tissue. So it's a very big hierarchy. We start with hematopoietic stem cells. Those cells are multipotential in the sense that they can give rise to all of the blood cells, lymphoid and myeloid. We start with neural stem cells in the sense that they can give rise to all the three different types of cells that you would find in the brain. But that's a non-trivial question. That's a very good question, actually, because it's very poorly defined right now.
Is the sorry, this is rather new to me, but
Good.
So the mechanism that the cell goes through when it actually differentiates, when the cell differentiates,
Right.
It looks like this is what you're trying to figure out. Like, how does this actually happen? How does it differentiate? So can you view this in real time, or at the speed that the cells differentiate, and examine it that way?
We can pick out the phenotype as it arises during differentiation. We can put in specific markers that allow us to know when certain genes have been turned on. Mm-hmm. You know, certain genes will be characteristic, for example, of a neuron. So you can put in GFP and then something that drives it that tells you when those genes are on. We're really trying to base it on phenotype and behavior. And how fast we can recognize it following a division, following the commitment. You know, what's the time lag between when it's definitely going to be one of those things and we can see that it's one of those things? That's one of the things we're definitely looking for. You know, how long do we have? What's our time window? I mean, one of the things that we play with in my lab is laser ablation. If we have a population of cells and we know that that one and that one and that one are ones we don't want, we can take the laser and go, pss, 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 and they're gone, right? I mean, we can vaporize cells with no problem. Right. So we can clean the population in real time, yeah. We can do that. And in fact, if you do it correctly, it won't even affect its neighbors because it'll basically shrivel up and fall in on itself. So that's where we're going. We want to do real-time control of all the cells in the population so that when I give you a sample, I hand something off to the doctor, I can put my stamp of approval on and say it hits all of these criteria. You know, and we've done enough testing in animals so that when these criteria are met, that animal lives a long, healthy life.
Other questions or comments? One, you're developing some technology for this as well. Could you describe that, what you're actually building as far as
Sure.
image recognition and so on?
It's very broad. I mean, we're really doing everything here. So I'll start at the start. So the first thing you need to be able to do here is long-term live cell imaging. So we've built incubators for microscopes. We've built environmental chambers for the microscope stage. We've worried about gas flow. Our patent is now under examination. We've developed a whole new culture chamber that allows us to grow most of these types of cells. Okay, so there's a system that we've developed that grows cells between parallel plates. That keeps them always in the focal plane, which is actually a very big deal because cells like to crawl over top of each other. And you can imagine that if cells is going over top of each other, tracking it becomes impossible. So what we've done is we've confined the cells to grow in a monolayer. So that patent is actually underway. It's in examination right now. So there's a lot of hardware we've developed. Okay, on the software side, we've really, as John will say, we were invoking feature creep. Essentially, the first thing was, okay, let's get our movies into some software so we can track it. And then, boy, wouldn't it be cool if they were colored? And boy, wouldn't it be cool if then we could speed it up and run it backwards? And then wouldn't it be cool if we could assign it? And essentially what we've been developing is a commercial application in C with a full Windows interface that my trackers can use. So there's in our lab, there's a cycle of development, use, development, expand, use, and it really is it's amazing to have someone in the lab that can implement ideas in the code very quickly. Because no one has really, lineage informatics is what we're calling this, and no one's really done that before. The people who do C. elegans work have tracked out every cell in a developing worm, but they haven't looked at it like this. In the worm, they know what every cell is going to be. In our system, we don't really know what everybody's going to turn out to be, so it's like an open-ended system. So we developed a ton of software. Now what we're doing is developing a lot of different ways to visualize the data as well. You know, overlaying information on a lineage tree allows you to retain the connectivity of the information and visualize where the properties are across that tree. And that's actually kind of a neat way to visualize this kind of data. So we're working on visualization approaches as well. So it's a lot of hardware, and really you can't buy on the market the hardware we've developed yet. You know, to get things so that it's stable over a long time, the temperature has to be really good, and the whole microscope is in our incubator. Okay, so you know we've got a whole bunch of foam boxes with $40,000 microscopes sitting in them in a little oven. Okay, taking pictures every 3 minutes, all the time. I'm in the process of ordering some hard drives for my lab. We do them now in 2 terabyte chunks. I'm about to buy 2 terabytes of hard drive. This is a lot of data. Okay, one experiment is several hundred megabyte. Okay. Because we can't compress things, because when you compress these images, they turn to mush. You know, we really need a pixel level accuracy here. So we don't want to do too much compression. Although there's some stuff that's outside of the cell area that we could compress. We haven't gone into that too much because a lot of the methods introduce motion artifacts. So we don't want to do that.
We actually have a group that's interested in the compression problem and forming around David Koff that might challenge that and maybe bring something to it where the compression could not damage the pixel level information that you're looking for. The question I wanted to ask, though, is what about the part of feature recognition? You said eventually you're going to be training a computer, as you said, but where are you going with regard to feature recognition so you can automatically follow these things?
Right. So that is done in collaboration with people in systems. Paul Figeth is a systems. So he has 1 or 2 students now. One of the students is simply working on automating the tracking. Okay, so, and it's tougher than it looks, because when a cell divides or when they do this dance or when they walk near each other, tracking, people can do that because they'll back up and they'll watch it and they'll figure out, that's because it's how we can hunt with a spear, right? If you couldn't track things, we'd all starve. But a computer to do that is tricky. So the computer has to form hypotheses about what it's seeing. And so one whole PhD project is simply on tracking those HSC cells and pulling out some features like, do they spin, what's their area, what's the nuclear size. So there's a lot of features that you can see. Another student is coming on stream, I believe, soon to start trying to program in what my student saw in her cocktail party. How do you parameterize a cocktail party or those sorts of behaviors? And I mean, this is a cognition problem, right? It's really a problem in psychology. As much as computer programming and stem cell biology. We need to be able to recognize behaviors in a way that's reproducible so that when we do our principal component analysis or our cluster analysis, we get meaningful clusters and reproducible. You're talking about medicine here, right? So the costs are kind of fuzzy. So really, we could have a room full of microscopes imaging every cell that we're planning on using for a transplant, and it might cost $100,000 to set up that facility, but big deal. That's nothing compared to the medicinal value eventually of those cells. I mean, if you could grow islets in a bioreactor for a patient, inject them into their liver so the pancreas regenerates within the liver, and cure diabetes, you would take $4.2 billion off of the Canadian government's expenditures every year, off the top. $4.2 billion a year. And that's just by getting rid of type 1 diabetes. Because it's the long-term complications that are the problem, right? So every time you come up with a new regenerative medicine approach, the dollar value is going to be huge, in the long term. So, sort of getting back to your question, I need an army of people watching cells and trying to parameterize these sorts of behaviors in a dynamic sense.
Have you looked at any of the work of the psychologists on that? In other words, they study the behavior of animal populations or mooses or something.
Children in a playground.
Children in a playground.
I did hire someone, and as a matter of fact, I hired a master's student, or she was an undergrad at the time, and she was doing childhood psychology, and I asked her to look at these movies and see if she could anthropomorphize this, but her brain couldn't get around that. It takes a pretty special person to be able to do this kind of stuff. It's pretty abstract.
It's interesting because the animal scientists do this all the time. I worked with a guy at Guelph at one point, and they were looking at the craziest little things in behavior, like how the deer turned its head when it reached down to eat something. But there may be people who are in that side of what they call ethnographic behavioral kind of
yeah, that's what we need. That's exactly what we need.
So when we talk to some of the people in that in those areas, we might find somebody.
I mean, your question highlights the point, though, that this is highly transdisciplinary, right? Engineers come into my lab and say it's too much biology. Biologists come into my lab and say it's too much engineering, right? You're telling me I really need an animal psychologist. You see the problem here, right? As soon as you start getting into a new area, as soon as you start looking at something in a new way, all of a sudden, you have to be an expert in 3 or 4 disciplines. And I firmly believe that all of the interesting stuff is actually happening at the interface between disciplines, right? In terms of chemical engineering, the Dankorot equation is done, right? Things get really interesting, though, when a chemical engineer meets a roboticist, meets a biologist, and then you start to get some new stuff. And so we are very, very transdisciplinary. And that's a bit of a problem for students. Because my students come in and I'm having them take biology, pattern recognition, process control, and reactor dynamics. What kind of degree is that? Well, it's a degree that allows you to start to do this.
Any other comments or questions? I met Eric, I guess not more than a year ago, and we tried to set up an imaging group and tried if it succeeded. And he came into the group, and I asked him what his department was, chemical engineering. I said, what do you want about imaging? And then he started telling me about all this stuff, which is wild in terms of its imaging implications. Luckily, there's a lot happening. Your data problem is being replicated now in every pathology laboratory in the world with whole slide imaging. We're talking about a half a gigabyte anyway per slide, maybe more, and talking about hospitals having imaging archives of 40 terabytes a year related to just pathology slides. Forget diagnostic imaging and so on. So there's new stuff happening here. Anyway, so I discovered that he's not only interested but knows a lot about it, and it's been exciting hearing about his stuff over time. The other thing is I know almost nothing about this stuff, and you made it totally clear to me, and I congratulate you on that. It was a really excellent talk. Thank you very much.
You were able to get, thanks. If you are interested, I am on campus, and you really, you can stop by my office and I'll show you some movies. Maybe email me ahead. Email ahead. I don't know if there. I think my email's on the first slide here. So if you do want to stop by and see this stuff, if you were brave enough to show up at this talk, then I will be happy to take the time to show you some movies. But you got to email ahead because it's grant writing time. And
but once he captures you, you're gone for at least a couple hours.
Yeah, right. Yeah, everyone likes to talk about the research. That's the problem with profs. So there's my email address. It's ericjj@cape.uwaterloo. And email me. You can set up an appointment. And I'll show you some movies. Because I'll tell you something. When I explain it to people, I figure it out. Right? If I can't explain it to someone, I don't have a clue what's going on. And so if you come over and ask me a question and I go, then I don't know. And then I can figure out what's going on. So, and it's transdisciplinary. So everyone comes in with a different question. And questions spark ideas. And, we're very into ideas right now, especially how to characterize and parameterize cell behaviors over time. Right? It's a big deal.
We'll look forward to that. Thank you all very much.
Thanks for coming up.